Virally induced vitamin D resistance, downregulation of the vitamin D receptor, Part 1. These are notes for June 5th, 2026. Virally induced vitamin D resistance is a clear example of acquired vitamin D resistance. We distinguish acquired vitamin D resistance from inherited vitamin D resistance, the latter being produced by mutations in the genome that result in defects in the vitamin D receptor signaling pathway. For instance, single nucleotide polymorphisms, SNPs, of the vitamin D receptor result in a vitamin D receptor which is weak or weakly activated, or in some cases, the SNP results in a weak expression of the vitamin D receptor. Now, this latter concept, weak expression of the vitamin D receptor, directly ties to the take-home message of this particular YouTube. We are going to be discussing how viruses, particularly herpes viruses, can impair the expression of the vitamin D receptor, the VDR. In this YouTube, we are going to be comparing effects of viruses on the vitamin D receptor signaling pathway. Two main groups of effects. One, the suppression of the vitamin D receptor, its expression, a downregulation of the vitamin D receptor. The second, which is more commonly discussed in the literature, is a suppression of the formation of hormones, ligands, for the vitamin D receptor. These are agonists that activate the vitamin D receptor, hydroxylated versions, metabolites of the inactive nutrient vitamin D. So, as a first example, we're going to be looking at human herpes virus 5, cytomegalovirus, abbreviated CMV. CMV is very pathogenic. It causes infected cells to form very large nuclei, which makes the cell dysfunctional. Approximately 70 to 90% of elderly humans are already infected with CMV, and yet the infection is generally not expressed. It is a dormant human herpes virus. However, with aging or other viral infections like COVID-2 or hospitalization, and particularly movement to the ICU, in these populations, CMV is more frequently expressed. What happens at the subcellular level? CMV is the largest in terms of proteins of all of the herpes viruses. It has more than 200 proteins encoded by its genome. When it infects a cell, it hijacks the cell's machinery, its organelles, its metabolism, and it also produces widespread epigenetic changes in the expression of host factors, host genes. And one of those factors is a transcription factor, SNAIL1. So CMV infections result in the upregulation of a host factor, SNAIL1, which has the deleterious action of suppressing another transcription factor, the vitamin D receptor, which is also a host factor. So in summary, a pathogen, CMV, causes a change in a host factor, SNAIL1, causing its upregulation. The consequence of that is an inhibition in the expression of the vitamin D receptor. This is an example of virally induced vitamin D resistance. Let me now describe how a vicious cycle can be established by the reactivation of the cytomegalovirus. At the bottom of the slide is a green highlighted information vector which shows a signaling pathway involving the vitamin D receptor, VDR. An agonist, 125D, which stands for 1,25 vitamin D. This could be either calcitriol, the kidney metabolite of vitamin D, 1,25 dihydroxyvitamin D3, or a therapeutic analog of calcitriol known as paracalcitol, 1,25 dihydroxyvitamin D2. To encompass both of these agonists, we can abbreviate them as 1,25D to refer to these doubly hydroxylated versions of a vitamin D backbone secosteroid molecule. That's a lot of verbiage, and the reason we need to define each of these terms is because we are going to be placing them together in a chain of causality, which is embodied in this information vector. So from reading left to right, an agonist, 1,25 vitamin D, interacts with the vitamin D receptor, activating it. The vitamin D receptor is a transcription factor. It upregulates the master anti-inflammatory transcription factor NRF2, which in turn upregulates a key antioxidant enzyme, GPX4, glutathione peroxidase 4. GPX4 reduces peroxidated lipids as well as reactive oxygen species. This is a chemical reduction of these oxidizing agents which create widespread cellular damage. Upregulation of GPX4 increases the cell's ability to withstand oxidative stress, which occurs during viral infections. One of the benefits of this upregulation of GPX4, it suppresses a form of programmed cell death known as ferroptosis, which is very prevalent in COVID and long COVID conditions. Ferroptosis is also a driver of inflammaging as well as other stressful conditions. So the medical intervention for vitamin D resistance is to increase the amount of agonists present in the body. And we have been discussing the most powerful immune hormone, which is the agent which maximally stimulates the vitamin D receptor, is calcitriol, 1,25 vitamin D3, which is highly synthesized in the proximal tubules epithelial cells in the kidneys. Calcitriol replacement therapy is also needed to suppress viral infections. And the vicious cycle that I would like to emphasize is that a COVID-2 infection can potentially reactivate CMV from its dormant state. The immune system normally suppresses CMV, but without adequate calcitriol in the serum or locally within specific tissues, the vitamin D receptors are not adequately stimulated, leading to immunodeficits, which could set the stage for the reactivation of CMV and other human herpes viruses. So what are the health associations with CMV reactivations? While largely dormant, the persistent immune activation from cytomegalovirus, CMV, has been studied for its association with frailty, cardiovascular disease, and increased mortality rates in older adults. Because it's well established that COVID-2 infections are more prevalent in older individuals and also immunocompromised individuals, which would include long COVID patients, the threat of herpes virus reactivations is very pertinent. And here we're discussing the ramifications of reactivation of CMV. The flow of information illustrated in this slide shows that in a tissue that does not have adequate access to calcitriol, this sets the stage for the reactivation of CMV, which locks in vitamin D resistance by upregulating a host factor, SNAIL1, which suppresses the expression of the vitamin D receptor. So this means the signaling pathway, which normally suppresses oxidative stress and by doing so suppresses a programmed cell death known as ferroptosis, without calcitriol, potentially you get the reactivation of cytomegalovirus, which locks in vitamin D resistance by suppressing the expression of vitamin D receptor. So this first slide emphasizes a host factor being downregulated in response to a viral infection, the host factor being the vitamin D receptor.